morning, students. Once again, we are going to continue with the sexual reproduction in flowering plant chapter. In that, I am going to start today about the post-fertilization events. So, in the post-fertilization events, what are the things we are going to study? So, the thing after fertilization, that is basically when the fertilization has taken place, there are some changes, what we call it as taking place during the fertilization, that is we call it as significance of fertilization. After fertilization, what are the things happening? So, those all things will be included here in the what we call it as post-fertilization event. That is, what are the things taking place here? The development of embryo from the zygote, then the development of endosperm. Endosperm is a nutritive tissue. Okay, nutritive tissue develops from pen. Pen is nothing but primary endosperm using this. Okay, then after fertilization, what are the things are taking place? These are the events we are going to see in the what we call it as post-fertilization events, that is the development of embryo from the zygote and the development of endosperm from the primary endosperm nucleus. Apart from that, the after fertilization, ovary is developing from the ovary is developing into the fruits, and ovules develops into seeds. Okay, then integuments develops integuments of the ovule. Integuments develops into fruit wall, that is testa and stegma. That is a seed coats, testa and stegma, that is stegma. So these are the events which are taking place immediately after the fertilization. The type of fertilization is double fertilization, triple fusion. In case of the what we call it as angiosperms, isn't it? So what are the things? That is, we after immediately fertilization, we have studied pre-fertilization event, we have studied fertilization event. After that, in sexual reproducing organism, which is the event taking place, that is post-fertilization event. So what are the things taking place in the post-fertilization events? I have written here, which in detail we are going to study in our today's class. Right? What are the things we got? Did you remember? That is one sperm that is one sperm that is combining with the egg cell forming the zygote, right? The sperm is a haploid, ovum is a haploid. So the zygote is diploid. This is a syngamy process in the double fertilization, which is the first fertilization. Right? Then one more sperm was there. That sperm combined with the polar nuclei, right? Polar nuclei to form primary endosperm nucleus. Okay, the sperm is haploid and the polar nuclei is diploid. So what about the primary endosperm nucleus? That is triploid. In all the angiosperm it is diploid, but in gymnosperm that is haploid. Why? Because there is no fertilization, it is going to be waste only there, isn't it? In the gymnosperm there is no sexual reproduction because the flowers are not available, right? At that time what happens? It is wasting. So that is the reason in the angiosperm, after fertilization only the endosperm is going to develop. See, what is going to happen? The zygote in future it is going to develop into embryo, right? Then primary endosperm nucleus is going to develop into endosperm. What is endosperm, beta? Endosperm is also 3N, that is a nucleated tissue. What about the embryo? Embryo is a diploid, that is 2N. Got my point? So here, what is a further feature? What is going to happen with the embryo? Embryo will undergo mitotic cell division and increases in number to form the new offspring. Right? What about the endosperm? It will also undergo the mitotic cell division. It means that it is maintaining the triploid condition. Right? And then it is going to serve as a nutritive tissue which is providing nourishment to the developing zygote. Got my point? So these are all, that is after immediately fertilization, that is the fertilization this is the second fertilization. We can call it as double fertilization and triple fusion. What it is called as? Double fertilization and triple fusion. How it is said to be, I said you people. I hope you people remember. Double fertilization and triple fusion. What do you mean by this triple fusion? Here, that is N, haploid combines with the diploid one to form a triploid. Primary endosperm useless, isn't it? So that is triple fusion. Double fertilization means first fertilization, second fertilization. This is one fertilization, this is another fertilization. Two fertilization. Double fertilization, triple fusion. Immediately after fertilization, we find these things. That is comes under the post-fertilization events, which are, that is forming the different structures and events I have written on the board. Okay, beta? This too you people have understood, or I have written already there. Apart from that, ovary, that is a female reproductive part, immediately fertilization hote hi. So flower loses the calyx and corolla. Calyx and corolla, that is petals and sepals. But in some of the conditions, the calyx remains along with the fruit. For example, in the brinjal. Brinjal में calyx रहता है. Calyx will not be disappeared. Okay? Got my point? Immediately fertilization होते ही corolla, that is petals, will disappear. It is degenerate or fall down, whatever it may be. Simultaneously, calyx also disappears or degenerate or fall, whatever. But in some of the cases, we find the calyx is remaining till it develops into fruit. Example in brinjal. Brinjal you will be seeing it is present, attached to the fruit. Fruit को attach रहता है ना brinjal को, green color का, that is a calyx. You don't find such thing in other fruits. Right? So ovary is going to develop into the fruit. Ovary will bulge, increases. You can easily see in the guava and pomegranate. क्योंकि फ्लावर ऐसा रहता है, ओवरी ऐसे बल्ज होता रहता है, आपको कब दिखाई देता ना, फ्रंट में वो है. So ovary will develop into fruit, whereas the inside the ovary ovules will be present. Ovules are going to develop into the seeds, and the integuments present in the ovule, integuments to integuments are there na? So those integuments develop into seed coats, that is testa and tegma. Two seed coats are there. If you soak the seeds, you can see the coatings. स्प्राउट्स में देखते हैं ना कोट निकल जाता है, ड्राई सीड्स में आपको दिखाई नहीं देता है. सीड कोट्स, टेस्टा एंड टेग्मा, 2 layers will be there. That is developed from the integument. Okay, clear? So now let me explain you very detail about the endosperm, embryo, seed, everything. Okay? Shall I repeat? So here now I will tell about the endosperm. Okay, endosperm. What exactly the endosperm? I will be telling you people. First one. Okay. Now let us discuss one by one structures in detail. So first I will explain better about endosperm. What do you mean by endosperm? Endosperm, which is triploid, but in gymnosperm it is haploid only. In NEET and CET, to make you confused, always endosperms are 3N triploid condition. We know in gymnosperms it is N, but it is waste then. It is not going to utilize in some cases. So that is the reason here after fertilization endosperm is going to develop, which is triploid. In gymnosperm it is haploid. Okay, shall I have these 2 points?
Yes.
So, endosperm is a common nutritive tissue, okay, where it is developing in the, what we call it as it is a nutritive tissue in the angiosperm. Okay, in the angiosperm. Listen here. एक क्वेश्चन फिक्स है यहाँ पे थियरी बोर्ड में, पीयू बोर्ड में, 1 mark, 2 mark और 3 mark. How? If they will ask in the 1 mark question, what is endosperm? It is highly nutritive tissue present in the angiosperm to provide the nutrition to the developing embryos. Okay, or else it is a highly nutritive tissue which is providing nutrition to the developing embryo. 2 mark में पूछा है तो what is endosperm? Write the types of endosperms. I'll explain you. 3 types of endosperms are there. Okay, अगर 3 mark में पूछा तो आपको वो टाइप्स के बारे में भी लिखना है विद एग्जाम्पल के साथ. ओके बेटा, shall I repeat completely? So it is highly nutritive tissue in the angiosperm which is formed by the fusion of what? Sperm and polar nuclei, right? During the fertilization, it has been developed, isn't it? Endosperm, if you talk about the endosperm, there are 3 types of endosperms. 3 types of endosperms. Okay, beta? Now, what are those 3 types, ma? First, nuclear endosperm. Then second is cellular endosperm. Then third is halobial endosperm. Right? So there are 3 different types of endosperms. You can see that one is a nuclear endosperm. Okay, nuclear endosperm, that is a coconut kernel. I'll tell you what are those. Okay. Okay, hello, nuclear endosperm, cellular endosperm, then one more is a halobial. Example, asphodilus. Asphodilus. Okay, so now I will explain you, my dear students. Shall I have that? I'll show you the diagrams. They will ask the diagrams also sometimes. They will see. This is what I am drawing is a cellular endosperm where only nuclear divisions are taking place continuously, which is not followed by the cytoplasmic division. Karyokinesis and cytokinesis are there. Karyokinesis and cytokinesis will be there. That is cytoplasmic division and nuclear division will be there. Cytoplasmic division and nuclear division will be there. So you can see here the three different types of endosperms we find in the immediately after the fertilization. You will be knowing that zygote also undergoing continuously mitotic cell division, and also what we call it as primary endosperm nucleus is also undergoing the continuous mitotic cell division, because they have to increase in size to develop into further structure. So the zygote will be undergoing the process of formation of the embryo. You are able to see here. It is at one end, it is at another end. Nucellus mein hai na? So you can see here, this is a cell, that is a polar endosperm nucleus. Polar nuclei will be there na? So this cell undergoing the process of cell division, only nuclear division. Nuclear free division, free nuclear division. यानी कि nucleus form होने के बाद they are separating, they are freely present. As you know that vacuole is present in the cell of the plant cell. Vacuoles are there na? So this is a vacuole. You are able to see here beta. Then many, that is thousands of free nucleus are produced by free mitotic cell division. Okay, it is seen in the tender coconut. Coconut pata hai na? Coconut water will be, tender coconut will be there, isn't it? So in that tender coconut we find this type of free nuclei. Later on, afterwards, these nuclei will be surrounded with the cell wall. Then they are going to develop into the milky tender coconut. Then milky tender ho jata, water ho jata. Then coconut completely develops, right? So that is an example here. So endosperm is here because embryo is here. Endosperm के अंदर है, देखिए वो. Have you seen in the coconut white color will be there small? Have you seen? So that is embodied in the endosperm. Highly nutritive endosperm. इसीलिए बोलते हैं ना tender coconut water we should drink. It is nutritive. Patients को दिया जाता है because nutrient content होता है क्योंकि endosperm है. Okay, it is a nutritive tissue, right, beta? So embryo is developing embodied in the endosperm. So which type of endosperm here? It is a nuclear, free nuclear भी बोल सकते हैं हम इसको, ठीक है? Then what about the second type? This is second type, that is a cellular endosperm. So free nuclear when you have seen in the coconut canal. Then what about the cellular adoxa? Adoxa is a type of variety in which the type of endosperm form after the fertilization. After the fertilization, so at that time what happens? Here that is cellular endosperm. See, immediately after fertilization a zygote is present, and this is a primary endosperm nucleus. So primary endosperm cell it is, inside that its nucleus. So this will undergo the division, cell division. How exactly? Equal furrowing. यहाँ पे देखिए zygote अपने आप डेवलप हो रहा है to form the embryo, and this cell is a polar nucleate pen, so it is undergoing to form the endosperm. So it is furrowing is taking place, as a result of which two cell stage we are going to see. Again, so many you are able to see the cellular endosperm adoxa. In the adoxa you can see this type of endosperms will be there. Then what about the other type? The other type of endosperm you can see, that is third type. Third type is halobial. In the halobial, this is the zygote and this is the polar endosperm cell. So it will undergo the process of cell division, as a result of which आधा यहाँ पे क्या होता रहता है? This is a what we call it as zygote. Sorry, embryo है, ये zygote है, developed into embryo, ठीक है? Embryo. So half it is undergoing free nuclear division, half it is undergoing cytoplasmic division. That is, cells are produced here half. So उसको हम क्या बोलते हैं? Cellular endosperm बोलते हैं ना? Then यहाँ पे क्या होता है? Free nuclear division. In the half, that is, it will divide it into two. In one cell, only nuclear divisions are taking place. So that is nuclear division, isn't it? And in half, in another cell, that is cellular division. Cells are formed now? Yes. So this type of endosperm is called as halobial. That is both cellular as well as free nuclear, isn't it? Both the types you are able to see. So endosperm is formed in many angiosperm, isn't it? Which do not only in few of the families, Orchidaceae family and Tripaceae family, there is no formation of the endosperm. Remaining all the angiosperm, endosperms are formed. Why? Because in order to nourish the embryo. If endosperm is not there, the embryo is not developed. If embryo is not developed, no reproduction. That is no development of the new offspring, isn't it? So one more question is going to arise here in this. There are, depending upon the type of the amount of endosperm produced present in the seed, the seeds are two types. That is albuminous seeds and non-albuminous. 2 mark का question रहता है यहाँ पे fixed. What, write the difference between albuginous and non-albuginous seeds. Okay, endosperm, when what we call it as endospermic and non-endospermic, albuginous and non-albuginous. Albuginous. Endospermic and non-endospermic. Okay, I'll write here. See, when the embryo is developing, it is utilizing the endosperm. Okay, so where the embryo is present in the seed, सीड में एम्ब्रियो रहता है. एम्ब्रियो डेवलप होते समय सीड में एम्ब्रियो भी रहता है, एंडोस्पर्म भी रहता है. सो व्हेन द सीड इज डेवलपिंग, दैट इज द एम्ब्रियो इज डेवलपिंग, इट इज टेकिंग द नरिशमेंट ऑफ द एंडोस्पर्म. सो ड्यूरिंग द डेवलपमेंट, दैट इज ड्यूरिंग द डेवलपमेंट ऑफ एम्ब्रियो, व्हाटएवर द क्या बोलते हैं उसको, endosperm is utilized. In some of the seeds, in some seeds, total endosperm is consumed during the development of the embryo. So there is no endosperm remaining in the seed. Whereas in some of the seeds, the endosperms are persist, yani remaining. Understood? Here there is no remaining, completely consumed by the embryo during the development. The seeds which are in some seeds, during the development of the seeds, the embryo is using, that is during the development of embryo seed, the embryo is consuming total endosperm, means seed में endosperm नहीं रहता, तो उस चीज को हम क्या बोलते हैं, non endospermic seeds. Okay, in some of the seeds, even though the embryo is developing by taking nourishment from the endosperm, at the end, okay, so depending upon the amount of endosperm consumed by the embryo during the development of the seed or embryo. So if the embryo is consuming total endosperm during its development, then do you find the endosperm in the seed after the development of the seed? No. Then there will be no endosperm remain after the development of the seed. That type of seeds are called as non-endospermic seed, non-albuginous antaradik. Okay, example pea groundnut beans. Adala developing embryo when it is developing. So when it is developing, in pea, groundnut and beans, the embryo of the pea, groundnut beans consuming total endosperm during its development. So in the seed, do you find any endosperm? No. Total endosperm will be consumed, will not be removed. So such type of seeds are called as non-endospermic seeds. Example these. There are some seeds during the development of the embryo, they are consuming the endosperm. But even though after the development of the embryo and the seed, there will be remaining or persistence of the endosperm, that is called as endospermic seeds. Example in coconut and castor. कोकोनट के ब्रेक किया तो उसमें एंडोस्पर्म रहता है ना, वाटर भी रहता है, कर्नल भी बना रहता है. ओके, we will continue the development of the embryo, seed and polyembryony and apomixis. That will be the end of the chapter. Any doubts you can ask me.